Welcome everybody, my name is Jim Hamilton. I'm the County Extension Director here for Watauga, based right upstairs. It's great to see so many different folks from so many different organizations and parts of the community here to talk about a topic that's very important for our area and historically has been important, and that's our forests and forest health. Dr. Dick McDonald, who's been sort of leading the charge for the promotion of using biological treatments for our hemlocks, will be here to sort of give you a big picture overview of what he's been involved with over the last 20 plus years in trying to protect and conserve and save our hemlocks. So we'll be inside for a minute, and then afterwards we're actually going to go outside and quote unquote meet the beetles. The beetles are active. He saw some yesterday and this morning while we were poking around, so we do have the Laricobius beetles in the hemlocks right around the Ag Center. He's going to demonstrate how to capture, catch, identify the beetles and their larvae. We're going to bring them back in here into our little ad hoc Dora the Explorer lab that we have set up here, so you can actually see up close and personal what the beetles look like, the larvae, recognize their excrement, because excrement is a neat ID tool for beetles amongst other wildlife too. Great way to ID whether or not you have the predator there. So after that, we have Blake Williams here. He's a program assistant for Ashe County Extension. So Blake and I did a survey, and we sampled for Laricobius across the county. He's going to share the results of that survey that we did in the fall. Jerry Moody and Lear Powell, who just walked in, are going to sort of talk about hemlock management and woolly adelgid management in the context of the landscape, as in for the landscaper's perspective. Because there's a different dynamic with hemlock woolly adelgid in someone's vacation property or second home, if there's a hemlock that's sort of a treasure tree on their property, compared to those hemlocks that are in the forest. And there's a level of protection that the predator beetles provide that's a little different than the traditional chemical treatment that we're used to doing. So that dynamic will be discussed as well. And then when we finish sort of the last round inside, for those of you who want to accompany Dick's going to be taking a group out to Grandfather Country Club, where the first major releases of the predator beetle took place, and you'll get to meet and speak with Mr. Gurdon, who was sort of instrumental in helping the predator releases gain sort of prominence as a treatment in these large POAs, property owners associations, that we have here. So got a lot planned, but we would like to make this as informal as possible. Please let us know if you have any questions along the way. If you have those questions, ask Dick, because he's the expert on this. But
no, Lear's here.
Yep, Lear Powell's here. Guest speaker. We're still waiting on Jerry. Jerry's not here yet. So for that, I'll introduce Dr. Rich McDonald.
Welcome everybody. Before I get started, I'll back up since Jerry Moody isn't here, and just give you a little bit of a background history lesson before I get going here. But I started working with Jerry, Jill, and Jeff, the Triple J's in Avery County, along with Doug Hundley. I had met Lear Powell, and we were all doing work on balsam woolly adelgid and looking at whether or not, if we modified the orchard floor management of these Christmas tree farms, if that would improve predation against balsam woolly adelgid, balsam twig aphid, elongate hemlock scale. And long story short, we did, but that stuff ended about 25 years ago. And I began to get into hemlock woolly adelgid. I'm a graduate of Virginia Tech. They had gotten this oddball beetle in their lab in 1997, and I was following this along and working with Fred Hain and Felton Hastings. And then in 2002, I got this phone call out of the blue from Stan Sturry. And Stan said, "Hey, I know that hemlock woolly adelgid is causing a big problem. Why don't we try and write, I know of a grant that we could write through the Urban Community Forestry Program at North Carolina in the Division of Forestry." Well, we wrote that grant and we got the grant. And the grant was to raise this beetle in the laboratory, this Laricobius beetle. And to tell you guys the truth, I wrote that grant without knowing what it took to raise this beetle. And then Gina, at that time her name was Gina Luker, and I traveled up to Virginia Tech, and within the first 15 minutes of me being up there, I realized I couldn't raise this thing in the lab. You have to keep the temperature below 73 degrees, and if you have a little tiny sprig of Adelgids, it's got to be changed 3 times a week because these beetles eat them all. And so suddenly I realized I couldn't do that, and I called Nancy up and I said, I need to change one word in that grant that you gave me. And she said, well, what's the word? And I said, I got to change it from laboratory to field. And she's like, well, that's a pretty big change. But we stuck with it, and now I'm just going to go into the story as it kind of unfolded from there, okay? And so if it wasn't for Stan and Carrie's understanding and Nancy Stairs, you guys, we wouldn't have taken the little tiny steps that got us going. And at the same time, I'm talking to Lear, I'm talking to Jerry Moody, I'm talking to other people about what is going on in this program. This guy, Leland Humble, was an impeccable forest entomologist, and he knew how good this beetle was. He was in Canada and British Columbia, and they used this beetle to control Hemlock woolly adelgid in that area. They knew how important of a predator it was. So he approached the Forest Service 2 different times and gave them this beetle. And the first time that he gave them to them, they had the same reaction that I did, because this thing comes out in October. This is the weirdest beetle that you've ever seen. And it's active all winter long. If the temperature gets to 73 degrees, it just falls out of the tree and goes down the needle duff and waits for it to cool back off. Okay? The very first thing that he told me when I met him at the invasive species meeting in Maryland, he comes up to me and says, you can use this beetle like a biopesticide. And I'm a biocontrol guy, and I'm leery of that. You know what I mean? You don't normally find beneficials that work as efficacious as this thing. At the end of me talking with him, the thing that he had told me was short term, treat your trees with chemicals, and long term, get as many of these beetles out as possible. Okay? Now, before we go any further, you guys know most of this, but this is what I would tell a crowd about. This is the thing that Lee Humble and the Canadians would tell me also, was trees that are off-site. So today, when we go outside and we look at these hemlocks that are up next to these buildings, those trees are off-site and they're stressed. They're almost permanently stressed. They're always going to have aphids and adelgids and leaf hoppers because they're not in the spot they ought to be. Hemlocks should be in a nice cove, have a very good root ball that's got lots of fungus and sawdust and needle duff, okay? And so when we go out here, you're going to be able to see both of those. You'll see some trees over here where they cut the tops of them off for power line and let them grow. Those that have a adelgid outbreak on them all the time because they're stressed out trees. They're off-site trees, okay? So once you realize that you have these 2 types of trees in an area, or at Hound Ears or at Grandfather Golf and Country Club, you can gently tell some people, because the best thing that you can do in some circumstances is take a chainsaw and cut that stuff down if it's really bad. So, okay. And the reason I put this slide up here is this is how this program started. I'm showing a picture of the world that we used to know 600 years ago. This is what the world looked like. That's all the world we knew. That's the same thing when we started with Hemlock woolly adelgid, right? We were told that the Hemlock woolly adelgid was native to Asia, it had no effective predators, and the hemlock forest was doomed. And that's one of the reasons I got in this program, because I said, well, maybe there's something that I can do. Maybe the hemlocks aren't totally doomed. And I committed to this program, okay? And so here's what I was just saying is, if you go on Google right now and you type in Hemlock woolly adelgid, you're going to see that the Hemlock woolly adelgid is still native to Asia. There are no effective natural enemies. You're starting to see a little bit of that now. And that the hemlock forest was doomed. So from 1999, when I really started actively, up until 2007, for 7 years, we watched the hemlock forest die. And then suddenly, we got a piece of information. I have 3 matrix moments in this program, okay? That's what I would call them, okay? They're all right here. The first one was, they did the DNA on the adelgid in the Pacific Northwest, and it turns out that that thing is native to the Pacific Northwest. and that it has effective natural enemies, and there are hundreds of eastern and Carolina hemlocks growing all over the Puget Sound, never been sprayed, as a living testimony to what happens if you put this beetle into a forest, what it will do, okay? The other thing that we realize is hemlocks get sun in the winter. On a day like today or any time in the winter that it's above freezing or actually above about 28 degrees, hemlocks are getting sun. They're not, how do you think the hemlocks are out here under this canopy out here? They don't compete when the leaves are out. They wait till the leaves drop, they turn on and get all that carbohydrate. That's why the Adelgid is out here right now trying to block the uptake of that. And this Adelgid, this generation of it lasts for 8 months, so it's really important. If we can use this beetle, and this beetle liberates the tree, every time this beetle eats 6 to 8 Adelgids every day, drinking their blood, every one of those needles turns back on and gives the food back into the tree. That became the really important thing that we began to see when we would put these beetles out, is all of a sudden within the next season, these trees that we had put, or shrubs that we had put beetles on, had leaders on them a foot long, because suddenly they were getting all this carbohydrate back, okay? And the predator, of course, was this Laricobius nigrinus, this crazy winter active predator. So we started moving beetles. I'll give you a long story short. Us folks in the mountain, we don't like to wait in line, especially if our trees are dying, so we quit depending on other people to get our beetles. And I personally made 90 trips to Seattle. I know the homeless people in the parks. I know who the ranking is, who to go talk to. And so we started moving beetles from that Blue Star area, Puget Sound, and we put them into our area over here. And this was the big game changer for us, because by 2006, we knew this Adelgid was native and we knew this predator was here. So we could turn on a dime and start getting this beetle, where larger organizations won't do that. And I want to emphasize this again. This is a paper. Anthony LeBoud is the one that showed me this paper, by the way, down at Fletcher. Hemlocks photosynthesize in the winter. The other thing that we had figured out at Virginia Tech was what the threshold was. Once you got below about a third of the needles infested on a tree, they just grew normal. So we were figuring out what the thresholds were for going out and looking at these things to see if we can find out what level of beetle and Adelgid doesn't hurt the trees. And we figured that out, okay? Why are western hemlocks average 175 feet tall? I think it's because of this beetle. But maybe it's the genetics, but it's very interesting to see. So I'm showing you this picture because this is in 2006, and this is when we realized that we could control this Adelgid naturally with Laricobius beetles. Okay, now since that time, you guys know most of this area has been infested. But this was the game changer for us, and I want to show you the beetle, and this is what we'll talk about. We'll go outside here in a moment. This is a winter active predator. It comes out in October, right when the Adelgid breaks estivation, summer estivation. This thing is synced up with this Adelgid. And this beetle feeds on 6 to 8 Adelgids a day. And really what it's doing is it's prepping itself for that cold period in the winter where it's going to get a whole bunch of fat bodies, and then we might have what we had this time where, you know, January and February is just cold as can be. We couldn't collect, there weren't beetles out because they'd all fed, and they just go down and hide in the needle duff and wait for it to get warm again. Once it gets warm again, okay, in the winter they're in sun tunnels out here because this faces west. We'll find beetles in all these hedges out here and over on these trees over here. Once it warms up like it is right now, they don't care about the temperature anymore, so all the hemlocks on the north side of Grandfather and north side of Howard's Knob and that whole area, all those beetles are over there now, tearing up the Adelgids and laying eggs. And so we'll see these eggs in there. And so one of the first things that we did, the very first week that I was in Seattle, David Mauzell and I dissected 1,200 ovis sacs and opened them up to see whether there was a Laricobius in them, how many eggs there were, and what the predation rate was, okay? And so what I want to tell you, all you got to focus on here is just this bottom bolded line where we had 50% of the ovis sacs were disturbed and 47% of them were eaten by predators. So we had 50% of them had an egg in it, okay? So if you put that together, you can put 50% together and 47%. Right away, we got a 97% predation of adelgids. And this was like, I want to see what's going to happen the next year in the very spot where we had these things. Okay, so here's what an egg looks like. We may see this today, by the way. We'll get a scope out and we'll go clip some stuff. We will see larvae and we will see adults, as far as I know. So the adelgid eggs are these burgundy color things down at the bottom, and that Laricobius egg is bright, big, yellow, and under ultraviolet light it glows bright green. So you can see all this stuff. When you put ultraviolet light on this, everything comes out and becomes that last matrix moment that I told you about, where we can find out what's going on. So what happens, a female beetle will lay an egg in an ovisac, okay? Now I'll go over here just for a second. Out west, the adelgid ovisacs average 175 eggs per ovisac, which is huge. You know what I mean? They are big, they are healthy, these are big trees. It's the Pacific Northwest, everything's a little bigger. So every larva has to eat 220 to 250 eggs or crawlers. Okay, so I average that out to 235. Every female beetle lays 400 eggs. Every larva eats an average of 235 adelgid eggs or crawlers. So one female almost kills 94, or about 94,000 adelgids dying. Now, out west, all they have to do is go from one ovisac to another to get 235. When we were doing our study at Grandfather, because it was so cold, we only had 24 eggs per ovisac. So that meant those beetles were laying an egg, and then they would count a dozen and lay another one. So they knew they never overstressed their larvae. And so what that would mean is if we're dissecting and we only get 1/10 of ovisacs with an egg or a larva in it, we got 100% predation. And so that's one of the things that we can do today, is we can count to see how many eggs are in an ovisac. Normally we do about 50. And then we can come up with that egg number, and then we can get our predation rate off of that, okay? So here we are back in Seattle. I want to show you guys this. This is the very limb. I actually hand counted all the adelgids on this limb along with David Mauzell. So we had 486 ovisacs on this limb, and that's one of the ones we had in our study site. The very next year, there were 4 ovisacs on that limb. The predators had knocked that thing down by 99.9% to the point where just enough adelgid. You can see there's an adelgid there, there's an adelgid there, and that starts the whole cycle over again, but it's so sparse that the beetles aren't interested in it except just to kill an adelgid. If they happen to walk by and see one, they're going to eat it and kill it, but they won't lay eggs on that. So this process of going from this to an outbreak in the city in Seattle takes 5 to 8 years. In a forest, it never happens. Once these beetles once a forest goes back into balance, it'll happen if you have a lightning strike or a drought or tornado or hurricane where you've got stuff that's stressed. But other than that, in the areas where we've put these things out into forest, and you'll see that at Grandfather Golf and Country Club today, it's been since 2007 since pesticides were applied on any trees of any size in there, and they all look great because they have these beetles on there. And so this just became a game of numbers. It wasn't about eradicating this adelgid, it was about knocking the number down to the point that it didn't affect the trees anymore. Okay? So this is the next part, and this is what we're going to do outside, but I'm just going to walk through it really quickly here, show you how we did this, how we collected and redistributed this, because this is now an industry that we can promote to the rest of the East Coast, because we've put these beetles out into all 7 counties of the high country. I don't care if you're at West Jefferson behind the Bantam Chef back there where the hemlocks are back there, there's beetles there. There's beetles on Roundabout Road, there's beetles in Sims Pond, there's beetles in Beach Bog. These guys will talk about that in a minute.